So I'm going to talk about gastric intestinal metaplasia, and actually hearkening back that one of my colleagues here talked to me about this nearly a decade ago. Lo and behold, here I am, and hopefully I have some things that will be of interest. So general long-term objectives is really to study the cellular and molecular features of gastric, and particularly high-risk gastric intestinal metaplasia, or gastric IM, which may lead to development of molecular or cellular classifiers to identify or consider high-risk lesions. Here's an overview of the talk, 5 basic sections. Well, you know, everyone's seen this, so we know this pretty much, so I'm going to skip this. We know this, but I think the point I'd like to make with this particular slide is that intestinal metaplasia actually is a series of cells, a series of aberrant cellular architecture that all interact with each other. And so learning to recognize what these cellular features are on a much more discriminate molecular level might prove to be extraordinarily informative, and I hope to convince you that we have found something along those lines. Olgam is something that this group knows much more about than I do, coming from med-onc background, but it does represent a relatively well-vetted, verified method of risk consideration of gastric metaplasia, intestinal metaplasia. And so this is the system, working with my gastroenterology colleagues, including Robert and Juha, that we've used to do some risk classification on our analyses. Okay, the concept I really want to bring forward here is this thought about integrative cellular genomics, where we use a variety of different platforms, each having their own advantages, and use this rather than as a single one-off, try to integrate all these data sets, which include conventional RNA-seq, of which there's enormous amounts of data available, single-cell RNA sequencing, which is a relatively new platform that allows us to discriminate cellular genomic changes at single-cell resolution, and spatial transcriptomics, which over the last couple of years has been a topic of a lot of interest, generally both in biotech and increasingly in use in academia. So the first component of this study was looking at general conventional RNA-seq analysis across gastric intestinal metaplasia. And through the gastric precursor cohort study, which was established by Juha and Robert, we had the advantage of getting a series of samples which were of high quality, and we could do conventional RNA-seq on these biopsies. Notably, these had been done along Sydney protocol, and thus we had Olgam staging information available. So from our first preliminary study, we had 88 biopsies from 46 patients. And just as a general classification, in order to do a supervised comparative analysis, we took all of these samples and basically broke them into a low-risk versus a high-risk analysis study. You can see the numbers here pretty much. And what was done, in particular through Ignacio Wichman, who is working with a number of people, including Robert, he managed to identify actually a very explicit gene expression signature that was associated with the higher risk Olgam stages, and importantly was independent of location, whether it was in the antrum or the body. So again, this was agnostic to at least those gastric locations that were identified. And so among this, there was a particularly interesting set of genes, as denoted by the red box around there, where expression was consistently elevated and found to be consistently elevated in the high-risk gastric IMs, and this occurred regardless of whether it was in the antrum or the body. So there are ways to take this kind of conventional, what they call bulk RNA-seq data, in which you extract all this RNA and you lose some of the cellular features, but regardless, you can actually extricate out some of these cellular features, and that's something that Ignacio actually did, in which he was able to recognize aberrant cell populations that are not typically seen in the gastric epithelium or gastric tissue, which included enterocytes, goblet cells, and intestinal stem cells. So the hunt was afoot. The next technique that we used as part of this integrative method was this spatial transcriptomics method. This was analysis conducted by Nuja Satra and Andrew Su, who also worked with some of the GAP samples provided through Juha and Robert. And this technique involves taking individual sections, H&E sections or solid tissue or flash frozen sections, in which you place them on a coordinated grid, and you convert the cDNA and attach the grid coordinates as part of the sequencing library preparation. So you can see a demonstration here of this spatial transcriptomic slide. You place the sample on. The grid provides you with coordinate gene expression information. Thus, you can map expression to tissue topology, as shown here. And as a result, you can actually take H&E results, histology results, and overlay that with gene expression. So we had the benefit of working with a fabulous gastroenterology pathologist, Jean Shen at Stanford, who worked with us to do the next part of the study. So from this independent sample set of 5 patients, we went ahead and did this spatial transcriptomic analysis across degrees of metaplasia, as well as a sample which had mixed adenocarcinoma dysplasia and metaplasia. And you can see some of the description of the samples here. And so what we did practically is, now that we had this increased gene expression signature where the genes were overexpressed, which has some advantages in terms of spatial mapping, we could actually take this gene signature and directly see where it was expressed upon these metaplasia samples. So below you see an example where Jean so kindly annotated aspects or features of metaplasia from the gastric IM samples, as well as different cell types. And notably, if you looked at an enrichment score that Andrew generated, you can see the hotspots showed up in red and yellow, localizing exactly to where the metaplasia areas are. And Andrew and Ignacio have generated some nice statistics on this. Subsequently, we went ahead and looked at aspects regarding this particular increased expression data set, gene set, and found out that it was actually elevated based upon these pathology annotations per gene, in which metaplasia cells clearly showed this in a significant fashion and were enriched, as you can see in these particular histograms, compared to the pit and base cells. So again, very good spatial evidence that's localizing the score associated with high-risk OGMs to metaplasia-based regions of the tissue. So we went even further and we did a little bit more annotation, and what we could actually do is look at the individual gene components that were overexpressed and see whether they really mapped across the sample sets and all the various samples that we had. And thus we were able to even further refine this gene expression signature to an even more discrete set of genes in which we could positively localize the expression to metaplasia cell regions. And then we took some of that information regarding these localized, spatially confirmed overexpressed genes and see and determine whether they're similarly expressed in gastric cancer. So this is something that Ignacio worked with Andrew on and demonstrated that these genes, in fact, were found to be consistently increased within a subset of gastric cancers, most namely those with chromosomal instability features. So good evidence that these signature features aren't just indicators of aberrant cell types, but have a carryover effect that we find subsequently in the actual invasive adenocarcinoma cells. And then what we could do afterwards is we can do actual mapping here, and so this represents an annotation of some of the histopathology we have, where we could look at included genes versus excluded genes along a gene expression scale showed per this log scale. And so included genes notably did map to the metaplasia regions, as can be seen here, with the intense red indicating where they are, and those localized to metaplasia samples, as you can see here. So in addition, we could further demonstrate that this very highly informative overexpressed gene set did localize specifically to metaplasia. So these are examples of regions which Jean denoted, in which you can see per the very intense red dots that this gene signature on this reduced gene subset explicitly and specifically maps to metaplasia. And you can see this even at relatively high resolution, where these small dots shown on the spatial transcriptomics clustering map, you can even denote very small regions or portions of metaplasia cells. So we seem to have an expression marker signature that's very specific and gives us pretty good cellular resolution here based upon a spatial transcriptomics method. Then finally, just as an additional confirmation, we found that the signature did carry over into cancer. So this represents a patient who had dysplasia, had metaplasia, and actually had invasive carcinoma. And based upon the heat map where increasing yellow-red indicates expression, metaplasia is predominant, but you're finding this gene signature also expressed within cancer itself. So the final component is albeit as good as this spatial transcriptomics method has, it's not single-cell resolution, and portion of the community, which I don't necessarily agree with, feel that you have to have single-cell resolution for some reason unbeknownst to me. But we can practically do this. So we took advantage of both public data set as well as internal data sets we generated to look at a series of gastric samples that included non-atrophic gastritis, gastric intestinal metaplasia, and early gastric cancer, and combined all of this data together So this was a single cell analysis looking at the gene expression across these samples of individual cells. So what you're seeing here is a demonstration of the entire aggregate data set, and this is analysis done by Anuja Sata working with Sue Grimes. And in particular, she noted that you could see an association with these different states, including chronic atrophic gastritis, gastrointestinal metaplasia, and whatnot. Now, just as a little reality check, we know that there's a stem cell gene called OLMF4, been described in the literature many times, associated with both gastric cancer and gastrointestinal metaplasia. And if we looked at those individual cells that overexpressed that particular gene, we find that they clustered, they weren't present in chronic atrophic gastritis or others, but they did show up in gastrointestinal metaplasia and early gastric cancer, as shown on the violin plot on the right side. So we're on the right path. If we explicitly looked at some of the components of our gene signature, we again confirmed, just as Ignacio had observed, and Anuja and Sue found this, is that this particular signature occurred in stem-like cells, in gastrointestinal metaplasia, enterocytes, goblet cells, and Reg3A positive cells. So cross-confirmation across multiple platforms that this signature was, in fact, an indicator of intestinal metaplasia associated with high risk of being converted into gastric cancer. And we used enrichment scores, which he's shown here, and this is another plot that Anuja did that's displayed by Andrew as part of his helping to put the presentation together. Again, 3 cell types, if not more, that really demonstrate an enrichment within this particular overexpressed gene signature. Now, what we were able now to do is actually begin to use this information because we now had a gene signature specific to individual cell types, and we could go in and discriminate where those mapped across the metaplasia samples. So here's an example where you can see a general metaplasia sample that Gene has outlined, as shown on the leftmost side, and you look for the yellow. But now we can exactly place where the metaplastic stem cells and the enterocytes occur, and lo and behold, they're showing up in the metaplastic regions. And again, these are genes that are derived from this high-risk signature of higher stage Olgan. Now, perhaps one of the things that I'm fascinated with, and probably to my group members are probably sick of hearing me talk about this, is the genomic instability features of high-risk gastrointestinal metaplasias that presage gastric cancer. There's a fairly a number of papers really looking at mutations which don't really seem to do a very good job of discriminating high-risk lesions, but clearly there's a story coming out that large aneuploidy changes where you see chromosomal arm imbalances are much better indicators. And with single-cell data, we can in fact discriminate this quite accurately. So here's an example where we're looking at the goblet cells, stem-like cells, and enterocytes. And lo and behold, if you look at the nice red box that I've circled out for everyone, located on the x-axis are the individual chromosomes going from 1 through 22 in X and Y. And what I've nicely circled for everyone is notice that the red signal in 8q is indicative of a chromosomal arm imbalance. And it turns out that's been reported by at least 2 other groups, be associated with intestinal metaplasia of higher severity. So the game is afoot. We seem to have a major chromosomal arm imbalance that is associated with potentially higher risk lesions. So in summary, what I think I've tried, or what this team has tried to identify, is a specific gene signature that's very indicative for high-risk gastric cancer precursor lesions through integrated multiomics, and we are working on a validation on this on an expanded sample set. What's notable about this high-risk signature is it seems to be retained among invasive gastric cancers, most notably of the intestinal subtype, and indications that this is related to the molecular subtype of chromosomal instability. And what we may be on is to start considering whether this might prove to be a useful molecular cellular biomarker to delineate high-risk individuals on follow-up endoscopy and see if we can use this to improve screening or improve the surveillance frequency of these individuals. So anyways, just want to acknowledge this really fantastic team of investigators, many from my lab, others including Robert Wang, Miranda Schum, who was responsible for the gene expression data, Juha's generous support for the project and his vision, and Gene, who's been just a fabulous collaborator, and without which we could not move forward. And then all the various members of my group, and particularly to Andrew Sue, because I stole most of these slides from him. So here's our funding support, and I'd just like to note that we don't have an NCI grant explicitly on this, but I'm looking forward to trying to achieve that one of these days. So thank you.